Next up, we've got Nathan Rossen, who's going to talk about the crystallization of the viral terminase DNA binding domain in bacteriophage P21. So, Nathan, take it away.
All right. Thanks, Paul. Today I'm going to talk a little bit about the research I did on viral replication in the lab of Marcos Ortega over this past year, both during the school year and as a student researcher over the summer. To quickly review viral replication, on the left here you can see an electron micrograph of the lambda phage. It's composed of three main parts: tail fibers, which identify a suitable bacterial host; the tail, which binds to the cell membrane and is the conduit through which DNA travels from the third component, the capsid, into the cytosol. Once this occurs, viral proteins are produced and viral DNA is replicated. The protein and DNA come together to form complete virions, which are then released. The part of this process that we're most interested in is the assembly, in particular, the packaging of the viral DNA into a complete viral capsid. So, the viral DNA, when it's copied using via rolling transcription, takes the form of a concatemer. This is a long molecule of DNA which contains several viral genomes stacked end to end. This DNA is packaged into a procapsid, which is a dodecameric hollow structure that assembles from viral proteins in the cytosol. At one of its 12 vertices, it has an opening known as the pore or portal, through which DNA enters during packaging. Once the capsid has been filled, it contains a single genome and is ready to attach to the assembled tail. For this process to occur, first there needs to be a cut made at the start of one of the viral genomes on the concatemer. Then one copy of the genome needs to be translocated into the procapsid, and then another cut needs to be made, severing the remainder of the concatemer. These activities are all done by the terminase enzyme. The terminase enzyme is composed of two major subunits, the large and small terminase. The large terminase, labeled here as GP2, binds to the portal and is responsible for the DNA cutting and translocating activities of the enzyme. The small subunit, labeled here as GP1, attaches to the large subunit and is responsible for the site-specific DNA binding activity of the enzyme. So you can see down here, there's two major domains of this small terminase, the larger of which binds to the large terminase, and the smaller of which binds to the DNA. So, my project was to determine the structure of the small terminase DNA binding domain in bacteriophage P21 via protein crystallography. So, this is the first time that I've mentioned my virus of interest, P21, and I'm not going to say too much about it besides that it's very similar to the lambda phage that Spencer talked about and that I showed earlier. Bacteriophages like these only infect bacteria, but we are interested in them because they are structurally similar to many human viruses that cause diseases such as chickenpox, mononucleosis, herpes, and the common cold. By studying models like B21, we can better understand how human viruses replicate. While we'd love to know the structure of the full small terminase, unfortunately, the region responsible for binding the large terminase is highly hydrophobic, meaning that it's not soluble and therefore is really hard to work with. So instead, we studied two truncations of this protein, one comprising the first 55 amino acids, the DNA binding domain, and the other comprising the first 106 amino acids, containing the DNA binding domain as well as a section of the flexible linker region. And the technique that I used was protein crystallography. So what is protein crystallography? Well, X-ray crystallography is a technique used primarily in the physical sciences to determine the molecular composition and structure of a crystal. But biologists use this technique to determine the 3D structure of proteins by forcing them to conform into an ordered crystal lattice. The technique operates on the principle that as you shine an X-ray through a crystal, it creates a characteristic diffraction pattern based on the electron distribution within the crystal. Using this pattern, we can reconstruct a three-dimensional electron density map. And by combining this information with a known primary structure of a protein, we can determine the three-dimensional orientation of each amino acid. Unfortunately, I didn't get to do any of this. It turns out crystallizing a protein is harder than it sounds. There's a lot of steps that need to take place before you can get any sort of data. First, you need to get a cell to express a large quantity of your protein. Then, you need to isolate this protein from all the other cellular components. Third, you need to introduce this protein to conditions that will induce crystallization. Only then can you get X-ray diffraction data from this protein. So, to express my two truncations, I transformed competent E. coli cells with two plasmids containing the genes for my DNA binding domain and my 106 truncation. These plasmids also contain an expression vector containing the lac operator, which serves as kind of a genetic on-off switch, preventing the translation of my protein until I add a specific inducer compound. So I transformed these cells, and then I grew them up in 6-liter batches. I then induced the expression of my proteins by adding IPTG. After lysing these cells, I had a substance that was very similar to fresh snot, both in its color and consistency. And this contained all the lipids, nucleic acids, and endogenous proteins that were within the cell, along with my target protein. So to separate out these components, I used two major techniques. The first is ammonium sulfate precipitation. This sorts the components based on their solubility in a high salt concentration. This removed most of the snotty debris from my solution, but I still had a mixture of proteins. So I used fast protein liquid chromatography, which allowed me to separate these different proteins based on their charge, using a 6 milliliter ion-ion exchange column, and their size, using a S300 size exclusion column. So, once I had a pure protein solution, I needed to get it to crystallize. And so crystallization operates on the principle that at a certain concentration, protein will precipitate out of solution as a solid. Typically when this happens, the molecules come together in a really disorganized fashion, creating this agglomeration that's completely useless for X-ray crystallography. But if the concentration increases very slowly, the proteins have the opportunity to come together into a repeating ordered structure. This is known as nucleation. Then, as proteins continue to add on to this nucleation site, a crystal begins to grow. So, to induce this, structural biologists create a closed system containing two different volumes. A smaller one, which contains protein, as well as a low concentration of a precipitating agent, and a larger reservoir, which contains only a higher concentration of the precipitating agent. Over time, as these two volumes equalize in concentration, water leaves the protein drop, decreasing the volume of the drop and increasing the concentration. Because there are so many different precipitating agents that you can use, and because there's no real way to know which one is going to work with a given protein, I set up hundreds of these different closed systems and then waited to see if there were any signs of crystallization in any of those conditions. Once I saw some signs, I went to optimize these conditions by varying the different concentrations of the precipitating agents and hoping to hit that sweet spot that'll allow us to get a large-scale, well-ordered protein. So, what did I see? Well, in the 106 amino acid truncation, I didn't see any crystallization at all. But in the DNA binding domain, I saw a couple conditions that showed nanocrystallization, such as this. These are basically just, they haven't grown much larger than the nucleation site itself. And I tried to optimize on these conditions, but I wasn't able to grow anything bigger than just more nanocrystallization. So I was pretty much resigned to not having anything pretty to show you guys during this presentation. However, on February 18th, 2015, I discovered a large-scale crystal in a tray that I set on November 20th, 2014, which, mind you, is my birthday. So, this is my little birthday present to myself. This formation is known as a needle cluster, which is several one-dimensional crystals emanating from a single nucleation site. So naturally, I go to optimize on this crystal, but no matter what I do, I can't get anything to grow, even in the exact same conditions. So what's going on? I talked to Marcos, and we, or I should say he, concluded that the problem was with nucleation. Because we didn't see anything grow for 3 months, but then once it did appear, it grew very quickly. A whole bunch of needles came out of that nucleation site. It was nucleation that was the slow process. So the solution to this is a technique known as crystal seeding. This involves taking a protein crystal that you've already grown and crushing it up into even smaller fragments. You then introduce some of these fragments into an already equalized protein drop, where they can serve as preformed nucleation sites from which crystal growth can occur. So I did this, and only 3 days later, what I saw was the growth of quite a few of these needles. There was a whole ton of them. It's hard to get a good picture of a one-dimensional object in a three-dimensional space. But only after 3 days. So to reiterate, I started with no large-scale crystals, then I got crystals after 3 months, and then I got crystals after 3 days, which was a really huge improvement, and I was really happy with. So to look back, I was able to express both of my truncations. I was able to purify both of these truncations, and I was able to crystallize the DNA binding domain. I'm only giving myself a small check mark for this third bullet point because the crystals that I saw only grew substantially in one dimension. And the highest resolution X-ray diffraction data comes from crystals with substantial three-dimensional growth, like the one that Ansa showed us in her presentation on Tuesday, I think it was. So, what's the next step? Well, I'd like to get some X-ray diffraction data from the needles that I have. It may not be the highest resolution, but it should still be able to give us some information about how the proteins are packing together in the crystal lattice. Also, I think we should continue optimizing using crystal seeding. I was only ever able to get one condition to do that. I only had time to do this one before I had to write my paper. So who knows, maybe just altering the conditions a little bit could get us a really nice three-dimensional crystal. And then if this doesn't work, then we can try other truncations. Maybe a 70 amino acid truncation will pack better than the 55 or the 106. There's only really one way to find out. And at this point, I would like to open up the floor for questions.
Questions, really good.
So, is there something potentially about the structure of the protein that sort of causes it to form a crystal in just one dimension?
So
I don't know anything about this field.
It's really hard because the precipitate, I didn't mention this, I had to take this out because I was going too long, but so often these crystals actually use the precipitating agent as part of the crystal lattice. So it's like there's an infinite number of ways that these things can be stacking together. And so it's like, yeah, it's definitely a structure, a function of the structure, but like there's no real way to predict how it's going to pack. There's so many different crystal lattices that can form.
I think, especially based on what you just told Mary, I'm wondering about if you're crushing up other protein crystals and using them for nucleation, is it like an issue when you're going to do X-ray diffraction that those things are part of the crystal structure?
My response to that is that hopefully that you get enough growth from, hopefully the nucleation site is small enough compared to the growth that you see that you can just fire it at a part that is not at like the interface between two different structures.
And I know this is a really obvious question that you might, but I just want you to summarize. So when you have that crystal structure, what is it that you hope it will tell you? Like, what's the most significant thing you're hoping to get from the crystal structure?
So you can get the exact conformation of the protein in 3 dimensions. Using the diffraction data,
Yeah.
and then you input that into some software, and that gives you this 3-dimensional space. And you can use software. You can kind of fold the sequence into something that kind of fits the electron shell.
I guess what I really meant was, what are you hoping that the 3-dimensional structure will tell you? Yes,
yeah.
like yes.
Well, so just like Sammy showed us, you can develop drugs that, if you know the structure of a protein, you can develop drugs that maybe bind to the DNA binding site and prevent packaging, and then you totally stop replication.
Great. Any plans after?
I'm going to take a year or 2 off and then go to grad school, I think.
Okay. Great. Thanks. Reached the end of this first half of the afternoon session. I believe we have a